Today we will be going over the B cell development, how a stem cell from a bone marrow differentiate into a B cell that will go around the body looking for antigens. There are four traits of the B cell. They are part of our adaptive immunity. They produce antibodies. They mature in the bone marrow, and they have pre-programmed antigen-specific receptors, which means no two B cells are the same. There are two phases of the B cell development. There are the antigen-independent phase and the antigen-dependent phase. The antigen-independent phase begins in the bone marrow. This phase can be broken up into four different stages, starting with the pro-B cell, the pre-B cell, the immature B cell, and the mature B cell. The mature B cell can then leave the bone marrow and go to other parts of the body. It can be a marginal B cell, meaning they stay in the spleen, or it can be a follicular B cell, meaning they go to secondary lymph node organs in the body. The purpose of those two is to look for antigen to come in contact with. Once they come in contact with an antigen, they enter the antigen-dependent phase, which they can become plasma cells, which makes our antibodies, or memory cells, which provide a stronger antibody response upon re-exposure. We will start with the beginning of the B cell development, which is the pro-B cell. We're going to start with our hematopoietic stem cell. A bone marrow stromal cell is going to come in contact with the stem cell. This will signal the release of interleukin-7. This is what we call paracrine interaction, which is a form of cell-to-cell communication. The release of interleukin-7 is going to induce gene expression. In this case, it's going to initiate the production of heavy chain receptor, which is the beginning of forming the BCR. The official stage of pro-B cell begins with the heavy chain gene rearrangement. This can be something also called the VDJ recombination. The VDJ recombination consists of four different gene segments. The V region creates diverse antigen-specific binding site. The D region creates binding site for antibodies or T-cell receptor. The J region helps join the segments together. And then we have the constant region, which determines the antibody class such as IgM, IgD, IgA, IgG, and IgE. The VDJ recombination is completely random. This means the random splicing of the gene is what creates the unique B cell receptors. The first part of the recombination is the splicing of the D and J region. This will form the DJ segment. Next, the V region will get spliced and join the DJ segment. And then we will have the constant region that also gets spliced, and they will form the final gene segment, which is our VDJ segment. With the VDJ segment completed, that will form our heavy chain receptor. The heavy chain receptor needs to be functional. If it is not functional, it will restart the recombination and resplice the gene segment until we get a functional heavy chain receptor. The pre-B cell phase begins with a functional heavy chain receptor. In the pre-B cell phase, the B cell is going to have an accumulation of the heavy chain receptor. The heavy chain receptor will then move to the surface of the B cell and combine with the surrogate light chain. The pre-BCR will also appear on the B cell surface. It consists of Ig alpha and Ig beta. Several rounds of cell division of the same heavy chain receptor will also occur during the pre-B cell stage. Going back to the appearance of pre-BCR, this will kickstart the light chain gene rearrangement. The VJ recombination is going to have 3 gene segments instead of 4. What it does not have is the D region. There's still the V region, the J region, and the constant region. There is still random splicing in this recombination. The V region and the J region were spliced together and joined. Then the constant region gets spliced and form the final gene segment, which is the VJ segment. Same as the heavy chain, the light chain receptor needs to be functional. If not, then recombination will start over. There are two types of light chain genes. There's the kappa and the lambda. 80% of human immunoglobulins will have kappa, and 20% will have lambda. Initially, all B cells will take on the mu constant region, which is IgM. The last part of the pre-B cell stage is IgM replacing pre-BCR with the immunoglobulin. The appearance of the BCR on the B cell surface means the B cell is ready to enter the immature stage. Just a real quick close-up view on the immunoglobulin. The variable region will determine the antigen specificity of the B cell. Negative selection is an important process that occurs during the immature phase. The random splicing of the genes increases BCR response to self-antigens, meaning the B cell can potentially produce antibodies that attack their own cell. To avoid this, the B cell undergoes negative selection. This process can also be called central tolerance, which is the elimination of B cell with self-antigen receptors. For example, we have a B cell with a functional BCR that has self-antigen receptors. A self-antigen is going to bind to the BCR. This will initiate a negative signal that will induce apoptosis. It is said that 90% of B cells die during this stage. At the end of the immature stage, the B cell will get their CD markers. Those CD markers allows laboratory scientists to identify different types of B cells. An immature B cell that starts presenting CD21, CD40, MHC2 means it's ready to enter the mature stage. In the beginning of the mature stage, the B cell will make its way out of the bone marrow. The type of B cell depends on what location they migrate to. When they migrate to the spleen, they are known as marginal B cell. Remember, B cells express IgM at first. Marginal B cell will express high IgM and low IgD. This is because they need to respond quicker to blood-borne pathogens. Once the B cell comes in contact with the pathogen, it will start releasing antibodies. Production of the antibodies stops once microbes are eliminated. Another mature B cell type is the follicular B cell. They will have a higher IgD and a lower IgM. Reason being is because they are circulating around the body between secondary lymph nodes, meaning that they have a higher chance of binding to self-antigens. IgM helps with self-tolerance, minimizing chance of binding to self-antigens. Once the follicular B cell comes in contact with the pathogen, CD4 follicular helper cells will bind. The CD4 follicular helper cell help mount an immune response. They can also differentiate into memory B cells that help mount a stronger antibody response upon re-exposure. Once they come in contact with an antigen, the mature B cell enters the antigen-dependent phase. The mature B cell can differentiate into plasma cells or memory B cells. The differentiation depends on T helper cells and the binding of antigen. Typically, a B cell that has a stronger affinity to an antigen more likely will become a memory B cell. Plasma cells reside in the bone marrow. They have CD138 on their surface. They have a shorter lifespan. They have little to no BCR. And remember, they produce antibodies. As for the memory B cell, they reside in the lymphoid organs. They have a longer lifespan so they can respond quicker to re-exposure, and they have a higher BCR level. And that is all for B cell development. I have created a review chart to point out key details throughout the development and differentiation of the B cell. I hope you enjoyed this video. Let me know if you have any questions, comments, or concerns.